Welcome geniuses, I'm Genie, your best buddy for A-levels. In this channel, we'll bring you to explore the secret formula behind success.
For this video, I'm going to cover topic 11 immunity for AS, and I'm going to focus on learning outcome 11.2b related to how do we use hybridoma method for the production of monoclonal antibodies. So before we start, let's define monoclonal antibodies. So MAB, or monoclonal antibodies, is known as antibodies produced by single clone of plasma cell. So since the antibody comes from one particular clone of plasma cells, so all the antibodies produced will be identical. Therefore, they are capable of recognizing the same antigen. With this ability, we are making use of monoclonal antibody for diagnosis, and we also use it for treatment. So now we're going to go into how can we produce monoclonal antibodies in large scale. So how do we produce monoclonal antibodies? As we know that antibody is produced from plasma cells, so can we just grow plasma cells in a culture and extract antibodies? It is possible, but the problem here is plasma cells are short-lived. So even you can get a plasma cells that produce your desired antibody, they might not be producing the antibody for long. Then you'll be thinking, why not I'll just use B lymphocytes? B lymphocytes, yes, it is long-lived, but they can't produce antibody unless they have differentiate into plasma cells. So now we are in a dilemma. So what can we do? We need to make the plasma cells long-lived. So in order to do that, scientists came up with a method where they obtain the myeloma cells, in another word, which is also known as cancer cells, and fuse with the cell, which is plasma cell extracted from the mouse, and fuse them together, forming hybridoma cells. So hybridoma cell, in other words, it's defined as a fusion between a plasma cell and a cancer cell. Now I'm going into the detail of the method of production of monoclonal antibodies. So number one, to obtain plasma cells that produces your desired antibody, we need to inject the mouse with antigen. It can be either a mouse or small mammals and after the injection, we will wait for 2 to 3 weeks before you kill. Reason is we will want the immune response to take place. Therefore, plasma cells will be present. After immune response has taken place and the mouse is killed, desired plasma cell will be extracted from the spleen. And to separate the cells, usually we will use centrifugation. And after you have extracted the plasma cells, that's where we will grow the cells in a medium culture that consists of a fusing agent known as fusogen, or the specific chemical is also known as PEG, in short, known as polyethylene glycol. So what is the purpose of this particular chemical? It's meant to stimulate the fusion between a plasma cell and a cancer cell, because if not, the cells will not fuse on its own. Then the next step will be, how would you identify if the cells has already fused? That's where we need another special medium which consists of HAT. This HAT is a special nutrient that only hybridoma cells can survive as they can feed on it. So if the cells are a fusion, in this case shown in blue, which is known as the hybridoma cells, they will be able to survive. As for the cells that failed to fuse, such as the cancer cells, they will die. And the plasma cells only will also die. Now, on the culture medium, there will be more than one hybridoma cells. Do you think all the hybridoma cells are the desired ones? So we need to screen in order to tell. Therefore, we will separate the cells into a different wells and grow them separately before testing. So now, if I have these 3 hybridoma cells, so what can I do to identify which hybridoma cells is the desired one? We will need to screen. And the screening is done by adding the same antigen that was used to inject the mouse or the small mammal that will stimulate immune response. So if this particular hybridoma cells can respond, they will be able to produce monoclonal antibody. For the cells that cannot recognize this antigen, it means that this particular hybridoma cells is not the desired one. It could be just some plasma cells that the mouse has previously produced when they are being stimulated by other antigens, but it's not your desired monoclonal antibody. So after you have identified the specific hybridoma cells, the desired hybridoma cells will be extracted, purified, and cultured in a large scale using a fermenter. And all the bacterial cells will be clones and producing the same monoclonal antibody. And this antibody will be purified and is used for diagnosis purpose or treatment purpose. So let's refer to this past year, which is from May-June 2017, paper 21. Before they ask about monoclonal antibody, the question actually asks about the structure of antibodies. So why antibodies has a quaternary structure? This requires your understanding from chapter 2, where you need to be able to define quaternary structure. So as we know that quaternary structure is defined as a protein with 2 or more polypeptide chain. So from this, we can tell that antibodies actually have 1, 2 short polypeptide chain and 2 long polypeptide chains. So in total, they have 4 polypeptide chains. Therefore, it is known as quaternary structure. B1. Explain how the structure of the variable region of an antibody molecule is related to its function. So as we know that monoclonal antibody, each and every one of them varies because of the variable region. So this variable region is specific to each antibody as the shape is complementary each variable region is specific to its antigen as the shape of the antigen binding site, which is here, is complementary to the antigen. So it's somewhat like the enzyme active site has a complementary shape to the substrate, but in this case, we cannot use the word active site. It must be antigen binding site and is complementary to the antigen. And this is due to the sequence of amino acid that forms the variable region varies according to the R group. So when you have different sequence of amino acid, you have different R group and different interactions, therefore the antigen that they bind to will be very different. So for part C, outline how monoclonal antibodies are being produced. This will be the method that I've taught you just now, where the keywords will be, number one, we have to mention about the antigen is injected into the small mammal, or you can name the mammal, which is the mouse. And number two, we will say that immune response will be stimulated. And the main thing that we want here is plasma cells is produced. So once you have your plasma cells, which is extracted from the spleen, we will actually fuse it with the myeloma cells. Or if you feel that you can't remember, you can just mention that it will be fused with cancer cells, stimulated by fusogen. Or we can name the chemical PEG. And the next step will be hybridoma cells are produced. And we have to identify which plasma cells is the desired one. Therefore, we need a screening method. So the hybridoma cells are screened using the same antigen, and once you have identified and the desired hybridoma cells, will produce the desired antibody, known as monoclonal antibody. And this antibody needs to be produced in large scale using a fermenter. So antibody, or we can say the hybridoma cells, are produced in large scale by growing in a fermenter. If you wish to add the marking point for HAT medium, we can add it in the point 5, where we can say that hybridoma cells are formed and they will be able to survive in a HAT medium, as this particular medium only allows hybridoma cells to feed on and survive. The last part of this question actually asks for the use of monoclonal antibodies, but they emphasize on the advantages. So as we know that antibodies are very specific, so in this case, if we use it for diagnosis, number one, we will say that they are very specific. So if I'm using this particular test kit with this same antibody, they will always recognize the same antigen, recognize or detect the same antigen. This is actually a very interesting topic where there's actually much more use of monoclonal antibody and the treatment examples of it. So if you're interested, please do not hesitate to contact me for group or private tuition. Thank you.
That's all for today's video. If you're interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with one or two past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.